So in today's session, mostly we'll be covering medical image segmentation. So you all have downloaded the 3D slicer software and all the images. Very good. So we'll look at cell segmentation, lung segmentation, and you will get to try it out on a patient data yourself using the code that is being provided to you. So do you remember MedSam? It was discussed in lectures before. MedSam. Yeah. So it's a foundational model for slicing, medical image slicing. Then you can also train your own model, and then finally you can look at the applied side of machine learning, deep learning, which is image classification. So in this image classification, you would be there are two classes, LUAD and LUSC. So what is the full form of LUAD? Very good. Lung adenocarcinoma and lung squamous carcinoma. So these are two different types of lung cancer. So you can look at the WSI images, and you can do classification as well. So and finally we'll be introducing you to diffusion models. So I hope you have a fun session today. With that, I hand it over to Manak.
Let's start with any questions if you have from previous sessions, or if you are able to download 3D Slicer and the folders that I have shared, session 2, everything is downloaded. We're ready? Okay, great. I think you should have access to this folder. So let's start with cell segmentation. So just download this IPython notebook or open with Google Colab. So I'll just open my personal Google Colab here. And okay, so for today's session, uh, you will need, like for almost all the notebooks, you will need to have GPU access. So so just to reiterate how to change your runtime type from CPU to GPU, you'll have to click the runtime here. Yeah, the file name is nitr underscore session2 underscore cell segmentation dot ipynb. And to change the runtime type, you'll have to click runtime, click change runtime type, and then click the T4 GPU. I hope it's already selected, but for people who somehow it's not selected, just click. Okay, so today, in today's lecture, we saw how Dr. Prasanna showed some histopathology slices where you have the cell segmented. So we'll, do a hands-on on how to actually segment the cells. I think somebody had a question, what's the difference between the normal whole slide image and the whole slide image with those green colors? So here we'll learn how to generate that. So uh, first we will install this GitHub repo, the cellpose.git. You can also go to their uh, GitHub repository and try to see all the things that they have. This was, I think, if I remember, like the first Cellforce paper was published in Nature Methods, and since then they had been updating their method. And Cellpose is able to segment different types of cells from across different stains and different cellular or different tissue types. So this is a good, I would say, segmentation algorithm, which you don't have to write too much code. If you have the histopathology image or whatever image that has cells in it, pre-process it a bit, and then you can use this code to generate your own segmentations. So yeah, today we will just see how to segment some basic cells. But if you want, if you are doing some project which requires some advanced input output operations, you can always take a look at the GitHub code. So given that we'll be running on Colab, so we don't have to necessarily worry about this. But in order to run the cell pose code on bigger images, you need to have a good system with some high RAM and high memory. And this is very well tested on Windows and Ubuntu, a bit less well tested on Mac OS. So I think you can try out on all the three different systems. Okay, So the first step is you'll have to run uh, this first step. So right now my session is connecting. It might take some time. It will take some time for you as well. So, yep. So one thing that I forgot to mention yesterday was if you write any code, for example, A equals 10, B equals 20, and print A plus B, and that will give you the result 30. So here if you see, I just start writing A equals 10, B equals 20, but here when I start this command pip install, yeah, the output is 30. But here when when I want to run this command pip install blah blah blah, I am putting this exclamation mark before that. Any idea why we are doing that?
So here using the bash terminal, that's all.
Exactly. So the cell remains the same. So this box is called a cell or a kernel, and here you are running a Python code, and here you are running a Bash code. But if you just add the exclamation in just before it, you can run both at the same time in the same editor. Well, this might seem very, easy and illogical to you, but believe me, few years back, if you are trying to do this, it will give thousands of errors and you will never be able to do it. But like now, given that people write bash commands, shell scripts, and Python commands and everything at the same, like they try to write everything in the same console or the same editor. That's why, people have come up with innovative methods like just adding an exclamation and you can run this. So I hope everybody has installed this Cellpose repo. Now we'll import some libraries. So yesterday we briefly discussed NumPy and Matplotlib, but and these two libraries we can discuss later, not important for deep learning task. But we installed Cellpose here in this command. Now it's installed in your system, but you'll have to call it. You'll have to call the function. So that's why in the second line we write from Cellpose import model, score, IO, and plot. And of course, I mean, you can still uh, run multiple other models in CPU, but Cellpose only supports GPU. So if you have not changed your runtime type to GPU, this will throw an error. So I will rerun this. It will just, so this line just loads the model and it waits from Cellpose. So you see the beauty here. You don't have to train Cellpose. You don't have to write the code for Cellpose. Just two lines. One line you import the library. In the second line you load the model and its weight. So you see like this 1.15 GB. This is basically the model and its weight. Now, just for visualization, we download some sample images, which is right there again shared by the Cellpose repo. Some cell images here. Let's download them and see. So it's downloading all the images, and then this matplotlib command will plot the cells. So you see the different cells here. I don't know what the cells exactly are, but people from biology background, can you explain what these different types of cells are? I mean, just to say I don't know the correct answer. So, anyone? So we just like from machine learning perspective, we have an image, we have the mask, we just want to segment it. Doesn't matter what the cells are, but if you're trying to do some downstream application, you need to know what the cells are. Even I need to know what the cells are if I'm planning to work with cells in the future. Okay, so these are the images. Let's try to plot some mask. So these are the images, and okay, we'll come to mask later because mask is basically the ground truth. Then I'm just feeding the images to model, and this model.eval. So there are two things in deep learning. Like we'll not do any deep learning coding here, but still just like hands-on if you are training deep learning models in the future. You declare a model. Let's say you made your own custom model, and you name it something like this. Let me write a pseudocode here. Model equals to model equals custom. model. And let's say you have a function. So you have a model, and I'm just writing flag equals 0, but let's say you have a deep learning model with multiple layer here. So here you call the model or declare the model, model equals custom model. So now in deep learning code, if you write model.train in PyTorch specifically, if you write model.train, I'm sorry, this means whatever happens after this step, like let's say, so whatever happens after step 6 till step 12, whatever you are doing with the model is getting trained. So let's say if you are passing through an image, you are passing the weights, back propagating the weights using a loss function. Whatever you are doing, the model is getting trained till model.eval. When you declare model.eval, that means the model stops training, and whatever happens after this stage, the model is just inferring. It freezes the weights. It is just doing inference. So I mean nothing to do with this code, but just quick insights. If you see a PyTorch code and if you see model.train and model.eval, you will immediately come to your mind. Model.train means whatever happening after this, the model is getting trained. Model.eval means okay, now the model is doing inference. It's not training it. The weights are frozen, so you can play with the model. So the same thing happens here. Model.eval, and you pass the images and use 1000 iterations, and it outputs something. We'll see what these output are. So if in the future you want to work with like cell segmentation, and if you have, let's say, a set of or a data set for which you want the cells to be segmented, you can just use the first few lines of the code, and your cell segmentation will be done. The most important part is how to visualize it, which will come after the inference is done. So we are almost at 54%. So just to state that the model is not getting trained here, it's just inferring. The weights are frozen. We are just doing inference here. There are multiple other cell segmentation methods as well. Like I think today morning, professor was discussing HoverNet, and I think there is another. Well, I forgot the name, but yeah, there are multiple other cell segmentation methods. HoverNet is one of the most popular one. So these models that we are talking about only does cell segmentation, but they still don't do cell classification, right? So Dr. Prasanna was talking about if this is a TIL, like is it a like a cancer cell, or is it a non-cancer cell? Is it a fat cell? Is it a lymphocyte? What is it? So to classify what this cell is, there are different algorithms. Those are cell classification algorithms. These are just segmenting the cells. It doesn't matter what the classes are. Any questions on cell segmentation? So are you guys confident if I give you another cell image, can you run the inference? Okay, great. Okay, now let's start visualizing. So here, just to keep in mind, whatever images will see from now on, the purple color is the ground truth, that is the actual cell, and the predicted is the yellow. So this ground truth might have come from a pathologist or a cell biologist. Okay. So let's see. So can you see the cells now? Like it's very well segmented using this yellow boundaries. So these are nuclei. So any idea why we can just see the so nucleus is inside the cell, right? but where is the cell here? Why we can just see the nuclei here and not the cell? Any idea from people with biology or whatever molecular biology background?
So we basically, I think, stained it through the through those stains, like which stains only nucleus, like DAPI uh, and rest, and the like it doesn't stain the cell membrane, so that's why it is not visible. So
Exactly. So if because of some staining we can't see the cell membranes or the other organelles of the cell, we can just see the nuclei, or we just want to see the nuclei, cell pose also segments the nuclei. So it's like, although we call it cell segmentation, but it can also segment the nuclei. And then it can segment the tissues. Then live cell. So live cell is a data set which basically has cells which are, like, moving in the petri dish or whatever the environment is uh, uh, across time. It can also segment these cells very accurately. Although just a quick glance, so this ground truth is basically in purple. So you see there is some errors here and there. This purple is basically the ground truth. So it has missed this thing entirely, it has missed some section here entirely, etc. Okay, yeast. So this is yeast, right? People from biology background? No? This, what cell is this? Yeast. So yeah, it's very densely located. Cell pose can also segment those cells accurately. And then the omnipose, the fluorescent, deep backs, etc. So people with biology background, you can actually play around with these data sets. They are publicly available for free. You don't have to, register or do anything. You can just like download them, train your own cell segmentation model using these uh, images, and then use it for downstream tasks. Okay, so now I will try to load a 3D image, 3D cell, and so basically cell pose works for both 2D and 3D cell structures. So somebody had a question in the morning that these are 3D cells. So yes, the images that we showed in the morning, they are from TCGA. So they are 2D but different magnifications. So the question can be, I mean, I had this question when I first uh, was trying to do cell segmentation. What's the application? Like, of course, Dr. Prasanna discussed a lot of applications after cell segmentation. You can create all those like graphs and extract features. What else? Okay. Let me simplify the question. So, uh, for live cell, the cell is moving, or the cell is growing. In order to understand the movement, or so what's the movement of cell called? I think the term is cytokinetics. Sorry, what? Motility or something. So in order to accurately measure, so I mean you can see with your visible eyes, but if you want to quantify it, put a number to it. Okay, what's the rate, motility rate, or movement rate or growth rate? There has to be a number. So in order to put a number, you have to quantify it. So the first step is the cell segmentation. Okay, so now in here what we do is we run cellpose 2D and stitch the masks because it's a big image. I think Dr. Prasanna did discuss stitching a bit yesterday, and I think you guys are familiar with that concept, so you understand in the back end what's happening. So you can use cellpose for that as well. And finally, these are, like, the cells divided across multiple patches. That cell post segments it and then stitches the mask. So for example, you see like this mask is here, but I think the patch just below it will have the other part of the mask. So you'll have to stitch the mask and then finally bring the main segmentation mask. And so that's it. You can do all the visualizations with these cells. Any question? It should be this one, right?
Yeah,
So does it say which line you are getting the error?
28, 29.
28, 29.
Yeah, outlines prediction.
Can
Name
you,
error.
sorry,
Name error.
aim error,
Name.
name. Okay, so did you run the previous, can you run it once again from the top? So that name error means you might not have declared a name before referencing it. Anyone else getting the same error? Like some error
No.
in this step?
Okay, okay.
No, right? It works? Okay, great. So name error basically means that you are trying to reference some name, but you have not declared it previously. For example, let's say I write a equals 10, b equals 20. So like this is cell segmentation plus Python tutorials. And then you try to write a column which will compute the sum of a and b. So def sum um, a comma b return a plus b, and then you just call sum, let's say. So it says missing two positional arguments a and b. But now, so that means you'll have to either reference the 10 and 20 here, so it will return an output, or you'll have to reference a and b. But what's the problem here? Let's say cx and y, and name this x and y. Sorry. So I'm not running this, but I'm still running this. So name x is not defined. That means it's the referencing error. So you'll have to first run it and then run this, and then the output will be 30. So this is the problem of this cell and kernel. This setup is called like Jupyter or kernel based compiler. If you are working on inline compilers, that means like you write the full code, have a python.py, and then run Python that .py file, you'll not have that error. It's just like you might misreference something in this Jupyter lab or Jupyter notebook setting. Okay, any other question for cell segmentation? What is IPYNB? This one? What? Where is that? Oh, after stitching. Oh, so this is just the like you can also name it anything. It's just a name. Like which plane I'm trying to stitch. I just named it I plane. You can name it anything like X, Y, Z. There is nothing technical or clinical in it. Okay. So the next thing is, what was the next topic? Lung segmentation. Okay. So we saw some CT images yesterday, right? So let me try to visualize it again. Okay, let me try to see if we have lung images in this one. No, there are no lung segmentations, but it's okay. We have some parts of the lung. Okay, so this has lung segmentation available. So now one of the like important application, or so for the first step is you want to get the lung segmented out for deep learning training or any downstreams. But another thing is, let's say if you have a tumor in the lung, you want to know which lobe it is in. So like any physiology, like or I don't know the exact term, like the branch of study that studies human anatomical organs, it's called physiology, right? Or human anatomy. Okay. So any anatomy guys, can you like name what are the 5 different lobes of a lung? It's very simple name. It's just like right upper, right middle, right lower. So it's nothing clinical there, but like yeah, you can reference it like this. So like it's very important to know where the tumor is located, because radiologist, when they get the CT, they find it there is a tumor, they would ideally like to write a note that the tumor is in the right upper lobe, or it's in the hilar region or something like, you know. So can we automate this process, segmenting the lobes of lung? It's very easy. So, so these are basically the lobes. You see, there are 3 lobes in the right lung and 2 lobes in the left lung. Okay, so we just install this library, pip install lung mask, and okay, before that, yeah, you'll have to upload the CT image. Okay, so my CT is uploaded, and hopefully my pip3 install lung mask also works. Also, just like try to have the runtime GPU. I hope it is. Otherwise, it will. Oh no, it's the CPU. So change the runtime type, save it. It will just kill the process. I'll have to upload the CT and everything again. So I think this will still run. What happened? Yes, I mean, so this library can still work on CPU, but it will be very slow. So it's better to use a GPU. So change the runtime type to GPU, and then install this library pip3 install lung mask. Uh, so if you want to know the GitHub repo for this code, lung mask GitHub, let's see if I can find it. Yeah, it's basically this. Don't worry, I'll send all these links after the sessions are done, along with the datasets and everything. So, so this is basically the GitHub repository for this lung segmentation code. So nothing fancy here. It's just a normal simple unit. Oh, before that, do you guys know what a unit is? No, no, no. Unit.
It is a segmentation architecture, sir.
It is a segmentation architecture, definitely. But and it's called a unit because it looks like U. I can just show you the the uh, the unit image. Yep. Is it visible? It's a bit blurry, right? But doesn't matter. You don't have to know what's written there. But it looks like an U, and the downside, like basically the descending part of the U is called the encoder, and then the ascending part of the U is called the decoder. So if you remember yesterday we discussed ResNet, we discussed DenseNet. So if you just cut the unit from this, like the bottommost part, the encoder is nothing but a ResNet or a DenseNet. And if you add another upsampling ResNet to a encoding ResNet, it becomes a unit. So very simple. So similar along the similar time when ResNet and DenseNet came, unit was published in Mikai, and I think in 2017 if I remember correctly, and uh, this model seemed to outperform or perform very accurately for segmentation tasks. So that's why like unit is famously known for segmentation. It's a very simple architecture, nothing fancy, but with time people just removed the convolutional based encoder and put a transformer based encoder, and that outperformed normal convolutional encoder, everything. But the unit, like for a segmentation architecture, this unit structure remains the same. We will try to look at a very advanced unit, which is, I would say, is one of the state of the art, the very famous state of the art, which you can readily use for your own segmentation purposes. So okay, coming back to lobe segmentation. So this is again a very simple unit trained with on this LTRC dataset. You can download this dataset, train your own unit, can be more accurate than this one. You can play around with this, but like if you don't want to train your own lung lobe segmentation algorithm, just use this library. And the code is very simple. It's just like we use this exclamation mark again because it's an inline or a bash command. Uh, write lung mask, then a space. Don't mess up with this space, otherwise you will get an error. And then similarly, copy the path and paste it here. Use inverted commas, otherwise you will get another error. And then like put any name. I just wrote lobe segmentation. You can name it X, Y, Z, A, B, C, anything. And then the model name is LTRC lobes because that is the data set that they trained on. You should use this if you want to output the lobes as well. And yep, just run it. I think it should not take much time in GPU, but let's see. So what it does is first it loads the model, then it reads the input, that is the CT image that we uploaded, and then it releases the it downloads the model path, and then it's running the inference now, and the output will be the lobe mask. Any question how to run this lung mask uh, uh, GitHub repo? So if you have a CT, mind it, you should be a CT image. So if you have a CT, you can just use this command or these two lines to generate your own lung mask. So meanwhile it's taking, like, while it's taking time, uh, can you, so this image we saw, okay, I'll come to that. Meanwhile, can you go to file, open a new notebook in Drive. So this is basically the third part, which is let's try. So we discussed cell segmentation, we discussed lung lobe segmentation. Now let's try the same lung segmentation algorithm on a new patient. So to do this, go to file, go to new notebook in Drive and click it. Let me know. I'll just take a break here so that we can, like you are seeing this, you are running the Google Colab notebook, which is already written by me, just pressing shift enter or those arrows, but let's try to do something on our own. So let me know if we have to stop. You, like, can you open the new notebook? It should say something untitled. For me, it's untitled 22. That means, like, this is my 22nd untitled notebook that I have. So here, like, you can either copy paste the same code, or basically, like, if you remember the code, because it's so easy, pip install lung mask and run it. Are you guys following, like opening the new notebook and everything, right? Everyone is following, right? We'll just take some time. Runtime, if you want to run it slower, you can use CPU, but I hope you will not, so change it to GPU. Yeah, click runtime, change runtime type, select T4 GPU, save. Yeah, and the next line of command is lung mask. So here what I would like you to do is, okay, let me also change the runtime type to just CPU. So upload this ts underscore ct. Can you see this? ts underscore ct. Just upload this. Although this does not have the full lung, but it will be a good experience to see what if we have just partial lung, how accurate this model is. And I am also doing it for the first time, so let's see. Like, I have also not tried on this CT. So upload the ts underscore ct. There in the drive, right? Just I'm trying to be clear. In the folder, it's not there. I think it's there. Can you try to re-download it? I have to refresh. So that means it's not there in my drive. Oh, after refresh it came. Ah, interesting. So I was willingly trying to not give you that that. Yeah, just download it. Like it should not take much time. And the code to run this lung mask is again an exclamation mark, lung mask, copy path, inverted commas, and paste. Don't forget the inverted commas. It will throw an error. So again, Python. Okay, I'll just give you some time. I'll not rush. Yeah. Yep, we are almost 51% there. So let's see. You guys are following, right? Like you can. Can you please repeat the error? What's happening? Which command? Not? Oh, did you install pip3 install lung mask? If says command not found, just rerun. Yeah. Or the spelling is wrong. Oh yeah. So the problem with pip libraries are, if you just make a spelling mistake, there will be another library with that same spelling mistake, and then you will think, yeah, it downloaded accurately and like it's not a problem, but then your actual code does not work. Yes, like I'm just waiting. Yeah, yeah, yeah. Yeah, so there should be some output file as well. So you can just name it like output 123.nii.gz. And more importantly, you need to have this thing, model name LTRC lobes, just after the destination path. Okay, let me just quickly check if my previous, okay, it's 72%. Anyone whose this segmentation is done? No, right? I mean, if it's not done for me, I'm hoping still there at some percent. Yeah, what's the error? Sorry, is there any error? No error, right? Okay. It's complete. Sorry, what? Lung segmentation completed? Okay. We'll just wait. Your computer is fast. Oh, sorry. LTRC lobes. Just try to see if the caps are in caps and small letters are in small letters. Is there space and everything? So what's the error? So did you? So is this model name correct? There are two hyphens. Yeah, my font can be different. So it's model, nothing fancy there. MODELNAME. It works, right? It's running. So I'm there at 94%. Like just a show of hands, how many like lung segmentation is done? It's done. 100%. 100%. Okay. How about you guys? Like your lung segmentation, the first one is done? Done? No, I'm by done I mean like the running this 94% is 100%, right, for you? Okay, for me it's 101%, so okay. So I have my lung mask with me now. It's stored here as lobe segmentation. I'll just wait for a few more minutes so that all of you have the first code done. The next step will be will visualize these lobes. We'll download these lobes and we'll visualize it. Yeah, yeah, yeah. So Fred and the first maybe the lecture of Python will be indentation error. You'll have to have space like in the correct places. You'll have to keep the capital and the small letters and the small letters. Don't start with numbers. Yes. And that is why I kept this live thing, because just running my code is easy. Writing with your own hands, even just these two lines, you will learn about how to write even basic codes. And even though running this is very easy, but let's say if you're doing this in your lab and you're not able to figuring out, and after multiple iterations, you're like, okay, let's scrap this. I hate deep learning. So yes. So those who have like already got this 100% done, you can just like uh, download it. So how to download it? Just click on these. I'll repeat it again. Just hover your mouse to this loop segmentation. You will see this three dots. Click that. You will get download. Any doubt? Any doubt? Are you able to follow? Yeah, I'm talking to you. Sorry. Yeah, yeah. No, it's okay. Yeah. Yes. Anyone who is not able to download or segmentation is not done, or there are any errors. It's running, right? Yeah, yeah, we'll wait. But any error in between? No, right? So for everyone, it's running. It's running. Which one? The second one? The second one, I will come back again. The live task. The live task, I will come again. But let's like first try to finish this download thing. So just like forget about the live task now. Just let's see let's see a show of hand for whom the segmentation running is done. It's not done? Little bit? Okay. How about you guys? Segmentation, that this thing at least is done? Done, right? Okay. So if it's done, click this three dots and click download. That's also done, right? Everybody has downloaded? Downloaded, right? Okay. Okay. So now open your 3D slicer. It might take some time to open 3. It's not opening? The last segmentation did not happen? Okay, my bad. Okay. We'll give you some time. Like, how is 3D slicer working for you guys? Like, can you open it easily, or it's taking time? For me, it takes time. I don't know. I'll have to throw away my laptop maybe. Uh, So upload. So okay, let me also close and open it again because my CT is already there, so I don't want. Yeah, see, my 3D slicer is broken. Okay, it works. Okay, so the first step is wherever you have downloaded this folder, uh, drag and drop this CT. I'm just repeating it again. Drag and drop the CT here. Just drag and drop. So in the folder you have the ct.nii.gz. Just drag and drop. Just check if it's volume, like if it says volume here. If it says volume and if the CT is the correct path, click OK. Everybody can open slicer, right? No problem in that. So drag and drop. Drag and drop is also done. So go to the folder that you downloaded. Are you able to follow? Sorry, last. Are you guys able to follow? Okay. Yes, do not open in Colab. It's a nifty file. It will not open in Colab. So drag and drop. Drag and drop is done. Drag and drop. Uploading the CT is done. Volume. ct.nii.gz should open as a volume. Okay. It should look like this. How about you guys? Can you open the CT? How about you guys? You can open the CT? No. So drag and drop the CT, and here it should be volume, and then click OK. Any problem with you? The image is different. ct.nii.gz. Did you scroll it a bit? Um, who is there? Okay, I'll maybe come there.
I have to open the output, no?
No, no, just the
Oh,
CT.
oh, okay, okay, okay.
Maybe close it and then open it. Yeah, please do not open the segmentation file, just the CT file. Any doubt? Like, can you open the CT? Right? Okay. How about you guys? You guys want it? Right now it works for you? Okay. Okay. So now, I hope you guys have downloaded the lobe segmentation as well, right? So wherever it is downloaded, for me it should ideally be in the downloads. I don't know where is it. It's not in my downloads. Oh, it's here. Okay. So again, same thing. Wherever you have your lobe segmentation, just drag and drop. But this time it should not be volume. It should be segmentation. So you have this description and then there is a drop down menu. Rather than volume, click segmentation. You can follow? Last. Yeah. Can follow, right? Yeah. Okay. And then click OK. So if you click OK, you can now see the lobe segmentation very accurately. Something like this. Can you see this? Like for everyone, it looks like this, right? No funny predictions. No, right? Okay, sometimes like so once I tried to run my own segmentation model in a very low resource setting, so I did not train it fully just to try out, and then the lobe segmentation was outside the lung. So Yeah. I mean, that's also a good learning, right? Like if you are segmenting lung lobe, it should never be outside the lung. So if you are trying to design a deep learning model, the first thing it should come to you, there should be some criterias which we call in deep learning as loss functions. You can penalize the model for predicting the lobe segmentation outside the lung, right? For example, the tumor, if you're trying to segment a tumor, you don't know where the tumor is, it should not be outside the CT. Bare minimum requirement is that. So, you can penalize the model that it should not be outside the CT space, it should not be outside the lung, something like that. But yeah, everyone can see this colorful lobe segmentation. You got a very what? Okay, let me see.
I
It's a
think
very
for me,
unique lobe.
segmentation is
Maybe
outside
it will be
the.
published in Nature. Let's see.
It went wrong.
Oh, I see. I see what you. Okay, let's close this. The same, right? This same colorful, old TV not working type of signals. Okay, I got it. What's happening? So whoever might have got this, weird lobe segmentation which looks like a cathode ray TV not working, you have to like, just okay. So for them, just close the 3D slicer and then reopen it. So it's step by step. First, upload the CT as volume. So did it open for you? Like now? Oh, yeah, it's opening. It takes some time. So first, upload the CT as volume. You closed and opened, right? You're already getting the error. No, I'm talking about, yeah. And then you uploaded the CT again. Now upload the lobe segmentation not as volume, emphasizing it, as segmentation. If you upload the label as volume on top of another volume, that is where you get this, uh, you know, I don't know what the term for that thing is, but I will you go go ahead with old TV not working type of segmentation. Okay, as segmentation. It's working for you now? No? Uploading. Okay, we'll see. Anyone getting error in lobe segmentation like this visualization? A very simple, well, you guys are in undergrad, we should not give homeworks, but what you can try to do is try to do the same thing in ITK Snap at home. You remember yesterday I was talking about another visualization tool, ITK Snap. You can try to do this, the same thing, upload the CT, upload the lobe segmentation and ITK Snap. So it's just like another visualization tool. Nothing else. So, like I think most of the tasks you can do using 3D slicer, but it's good to know other, uh, segmentation tools. Yes, it does, but like for basic level, it does not matter. Like I use these just for visualization tools, so they might have different set of interactive segmentation. So maybe 3D slicer, have some set of segmentation tool which are better for some set of radiologists, and then ITK Snap might have some other added functionalities which might be easy easy to use for some other set of radiologists, but we are neither of them, so we are just visualization guys. So just drag and upload, and yeah, this L, P and S value, right? That's a good question. What do you think it is? What do you think it is? What's LPS in CT? Okay, okay, see, just scroll, so drag your, put your cursor here and just scroll it. So what is changing and what is not changing? No, two values should change and one should remain stable. So you see like L and A is changing, and here the X and Y is changing and the Z is not changing. And then if you get your cursor here, the R and S is changing, P is stable, and here the X and Z is changing and Y is not changing. So it's basically which slice you are in. Does it work for you, person in the last? Like, You got the lobe segmentation like this? Everyone got the lobe segment. Did you download the CT and everything? It's helping you. Anyone else? How about you? It worked for you, right? I was helping you. It worked, yeah. So, like reiterating again, you'll have to upload the ct.nii.gz, not the ts underscore ct. Yeah, I think I should have kept that brats underscore 00000, that name. I just simplified it to ct and mr, and now and that creates a problem. It's okay, right? So everybody is at this step should have the tumor. Oh, sorry, not the tumor. The lobe segmentation on top of the lung, right? Everyone is here, right? Right? Okay. Because the next step is also very crucial. So you should be at least here. Everyone is here, right? Okay. So now what you can do is you can scroll through and see the different lobes. See, it's segmented very accurately. Okay. So now the next part is how to visualize this lobes in 3D. So now it's all 2D, right? Like I can see different planes, but like, well, I want to see the segmentation in 3D, like rotating in the space. So to do that, click Welcome to Slicer. This thing. I'll repeat it multiple times. Click Welcome to Slicer. Don't click anything else. I'll come back. Just click Welcome to Slicer, right? And can you see segmentations here? Don't click. Just like the movie. नहीं दबाना था. Sorry? Yeah, I have not clicked. So just here, just to show everyone that you'll have to click the segmentations. Everyone is here. Segmentations. Now you can click it. So everybody should see this thing. Segment 1, segment 2, segment 3, segment 4, segment 5. There are 5 lobes in a lung. If you're getting 4 or 6, that means your segmentation algorithm is wrong. Or sometimes maybe what happens is like you take the lung out. In that case, you might have less, different numbers. But for a normal lung, you should have these lobes. Big tumor, take a significant part out, and small cell lung cancers are generally very big. So, okay, so everybody is here. This segmentation 1, 2, 3. So you remember yesterday I was talking about like how you can change the opacity and stuff. So click the display. Everybody can see this pane, right? No? Can see, right? You can play around with opacity. So what radiologists generally do is, so in order to see, well, if the segmentation is accurate or not, they change the accuracy and see the soft tissue, the difference between the soft tissue and air, and the soft tissue and bone, and the air and bone, whatever the structure, depending on the anatomy, try to see how accurate the segmentation is. If you want to check the, so basically, for example, if I want to ask you segment the lung, you can still do a good job because difference between the soft tissue or the bone around the lung and the lung space, which is full of air, it's very simple to segment, right? So let's say you developed a lung segmentation algorithm and you want to check how accurate it is, you can just like, play around with the opacity. And then space fill as well. If you want to fully fill the space, or if you want to just like have contours, so you see like this contours, and if you want to like visual just like copy this and plot it in a presentation, you can do that. Okay, we'll just wait for a few more guys to complete this lobe segmentation and everything, and then we'll move to the 3D visualization part. Yes. So contour is it's nothing. It's just like you will have to take the space fill. I don't know what this scroll thing is called, and just put it to 0. and you will get this contours. So this is good for visualization because like if you report this in a paper or a project report, you can still see the contours and you will automatically know whatever is inside this contour is basically a label. So yeah. So like if so now I removed segment 4 and 5. Like generally your natural eyes will not be able to say what are the two different lobes. But can you see this line here? Very, maybe in your computer it will be better visible than this one. So you can see a line. See exactly that is where the model is contour or model is segmenting it. So that is like for natural eyes or for naked eyes or for inexperienced eyes, lobe segmentation can be like, well, I don't see any difference, but like once you see the segmentation you can still figure out, yeah, there is some difference between these two lobes. So as of now I will just fill these. Okay. And then if you click this show 3D, this button, just click it, nothing is there, so but still click it. So what's happening is the segmentation is, so you just see a small window, the segmentation is somewhere in some axis. To bring it to the center stage, go to this small pin to the leftmost button. So you see the small pin, and click this second center view icon. Great. Okay, I'll just click it. Now you can see the segmentation in the center view. I'll repeat it again. The small pin, the second icon. I hope it's the second icon for everyone, right? Yeah, okay. Okay. Everyone's there? Second icon, click, can see the segmentation now. No? So like segmentations are there, right, for you? And like what happens if you click the second icon? Oh, did you click show 3D by the way? Again? Okay. Okay. Everyone is still this step. Like can you see the slope segmentation? This is not it. So now like you see like this segmentation is not like a looks, this does not look like lung. So we'll have to change the view. So you see this 4 northwest east west kind of a thing. Click the A. Take your cursor to A, click A. Now you can see it looks like the lung that we generally see in images. I just scroll a bit, right? Click the A. Just click the A line. Done. Everyone can see the lung in the natural lung format. And if you click the second like 2 icon, like this one basically, it will spin just for interactive visualization. Now, so that is it for today. Like not for today, sorry. That is it for till I get the tea that is for me today. Uh, Stop the spin and then you can visualize the just one thing. If you have done this segmentation, you want to publish this or you put this on a report, how will you do that? Don't take screenshot. That is like a don't take screenshot and put in a report. You can take the picture but don't take a screenshot. So you see this eye option, click this eye option, remove the 3D cube, remove the axis and everything, just click this 2 icon and white background. So now we have the white background. Zoom till whatever level you want and then right click on it. So yes, repeating once again, click this eye icon. There is a white background. Let's change background whatever you want, like light blue or black or white. I just prefer white. So everybody is still this stage. Click this eye, remove the 3D cube, remove the 3D axis label, and have white background. Everybody is still this stage. Looks like normal human lung, right? Right click on it and just click copy image. And then if you open a presentation, here I'll do new slide, just remove this, and you can paste it here. So this is much better quality than a screenshot. The next thing that we'll try out, so I'll skip MedSam again for next day. MedSam, I don't know what MedSam has to, like the enmity with MedSam, I'm just postponing it. But I think total segmentator is something which you will find more interesting. So let's discuss that first, and then we will try to quickly do same, tutorial with total segmentator. Okay. So similarly, upload. I don't remember the file name. I think it's called nitr underscore session 2 underscore total segmentator, right? Let me know if you have this file uploaded, right? Everyone? So this upload the total segmentator file. The same procedure, file, upload, or right click and open in Google Colab. So I'll just repeat for the people who just joined. In the folder, you will have a IPython notebook named nitr underscore session 2 underscore total segmentator. So just open that in Google Colab, similar like how you did in the previous few, iterations. So let me try to give a very quick, background about total segmentator. Um, I think just few minutes. So I have a habit of uh, spending too much time on LinkedIn, and and like what papers are getting published, where my friends are getting their jobs, and which jobs are open and everything. So I came across this post, and I was like, so this is something. So this project was something that I thought of doing, like if there was enough data set, enough labels, and enough, training resource. And when I saw that this happened, rather than being sad, I was so overwhelmed. I just told Pratik that, see, this we were talking about, and this is now there out in public, and he was like, no, that's not true. And then I had to show him a demo that, yes, see, this works, and they have a visualization, or have a platform where you upload the CT image, and it will give you a segmentation. So what total segmentator does is it segments 108 anatomical organs, right from the skull to the toe, all the bones, the gastrointestinal tract, different organs, the muscles, different muscles, and the cardiovascular system, which has the aorta and all the other vessels, arteries, and everything. So basically, everything that you want. So as bioinformaticians, right, or medical technologists, or whatever you guys like to call yourself, like it starts with, as I kept saying, it starts with segmentation, and whatever organs you might want to work with, which is solid organs, like of course blood segmentation. I'm pretty sure it's not like a thing, but any organ that you want to segment, total segmentator kind of like does that on CTs, and now I think since last year or last few months also on MRIs. So if you have a CT, a full body CT, or even like whatever part of the body you have, total segmentator can segment all the organs for you. And again, the code is very simple. Pip install total segmentator, and similar to lung mask, yeah, it just downloads a lot of things. And exclamation total segmentator dash I, the path of the CT, and dash O, the segmentation CT. So let's do this. Let me run it. I'll have to change the runtime type to GPU, run it, and then again run it again here. I closed everything. So, oh, this one. No, this is the drive. I think it's closed. Yeah, I think it should work now. Let's see. So install. Where is that? Manage runtime, right? Manage sessions. Terminate other sessions. Yep. Close everything that is there. Okay. I think I just refresh once again. But are you guys able to do it? Hmm.
Showing a warning. The following packages are previously imported into runtime. However, you should restart.
Restart. So restart again.
We restart completely showing the same.
So don't do so after restarting, right? Don't run pip install total segmentator. Just start with the next step.
You know?
Yeah, don't. So once you run pip or pip install total segmentator and it like restarted your runtime, don't run it again. Just run the next line, which is not working for me. Can anyone help me? Another account? Yeah, I think this will work now, hopefully. No. Okay. Do I have another account? So I'll have to give access to myself. But let me try with manage sessions once again. Yeah, it says runtime disconnected. Yeah, let me try that. I'm just giving permission to myself now, so. So it's still running, so I'll just stick with this.
After reconnect, you have to again upload the image file in the data section. Okay. After reconnect, you have to upload again the data, like the data file. You have to again upload it. Okay, and then you can do it. Yeah. That file name is ct.nil.gz. We have to upload again.
So are you guys able to run and did all the segmentations downloaded? No, right? No? Yes? Downloaded. It's done? Is it done for you guys? Rest of you? Download like that the CT thing run for you guys? Everyone? Okay. Please wait for a minute, a few more minutes.
I think we can move ahead, okay? Like,
Yeah. So like a note, like total segmentator, although it segments all the anatomical organs in your body, but let's say if you have a CT which is just capturing the thoracic region, or if you have a CT that is just capturing the abdominal region, you can still like to use total segmentator to generate the segmentations only for those regions that you have. So we'll see how it works. Yep, it gave me an error. The input file does not exist because I never uploaded it. So meanwhile, just also upload the TSCT and the TSMR. TS stands for total segmentator. So now we are going to use that, which some of you were visualizing it by mistake. So for the guys who have already like for whom the segmentation is already done, just save that in your computer. So similar, like how to save, click that three dots, and then there will be a download thing. Just download it. It's not there. Oh, you can. I mean, so what you can do is you can zip it and upload. I'll share the code. 1 second. Or what you can do is try to take a look and see like if there are any segmentations like lang.nii or something like that. Download that first. Is there a lang.nii inside that folder? Everything is there, right? Just select like 3 4 segmentation, your choice, download them and try to visualize. So everyone were able to run the total segmentator, at least the first 2 lines.
After the segmentation part is generated,
Mhm.
then we have to download certain segments.
Yeah, just select the segmentation of your choice, lang or something like that. So meanwhile, open your 3D slicers again. Like close it first so that the previous lang and previous segmentations are not there, and then open 3D slicer again. Are you able to reopen the 3D slicer? Now it works fine. Yeah, for people like who have already done the CT, maybe it can also run the same thing for MRI. But for the MRI, do one thing. Don't use this MRI.zz. Just use the TS underscore MRI. Okay. So I'm repeating once again. So the people like who are already done with the CT segmentations, leave it there. Just start running uh, the same code. Just change the path from MRI to TS underscore MRI. MR, sorry, my bad. MR, and then run it. So for that you'll have to upload the TS underscore MR. So I'll also do the same. And yep. So just a rain check, like everybody has the segmentation done, right? So you should be able to see the segmentations underscore CT in the left data pane or in the files pane. Everybody has segmentations underscore CT, right? Right? Okay. So do something like just try to download something. For example, I'm trying to I'm downloading aota, and then okay, I'll just save it aota.nii, and then maybe the lung lower left. Yeah, I'll just try to like download these two labels. You can choose your own label. Just a note again, revisiting my note. So we are running this total segmentator on CT nii.gz. If the organ is not there in the CT, it will still predict a mask, but it will be a zero mask. So even if you drag and drop it here, there will be nothing. But let me see how accurately it predicted the aorta. So again, a quick reminder, you should not, like, while dragging and dropping the aorta.ni.gz, it's a segmentation file, change from volume to segmentation, and then click OK. So I can see the aorta now in green. Aorta, like medical, like people with medical background or anatomy background, what is the aorta? The microphone, the microphone to someone.
It is the largest valve from where the blood flows, enters the heart.
Okay, okay. The It's the main artery, the
Largest artery.
largest artery in the body. So that's what I was trying to say. Like, the blood does not flow inside. It sends the blood to the entire, to different part of the body. And it's a thick structure. I don't know if you have ever seen a real aorta or not. It's a very thick structure, and like with age, you develop something called abdominal aortic aneurysms. The aneurysm is basically, it will just, like, the wall started to thin, and it starts to bulge, and then calcium gets deposited, and if you are not like if you are lucky enough, you will diagnose it early. If you are not lucky enough, like, the aorta can rupture, and it can lead to complications. And interestingly, many important people have died because of the rupture of aorta. Can you name any famous one? A very famous scientist died of abdominal aortic rupture. Albert Einstein. Okay, so this is the aorta. Any problem uploading the aorta? No, right? No. The aorta is not visible. Wait, the green. Oh, it's not visible here. Okay, let me. Or it's not visible for you. Here it's not visible, right? That is not a problem. We just learned how to do this display and everything. How about now? Still not visible? That is the problem with the light. You can see the green segmentation here, right? You can see, right? Okay. So let me try out with the another segmentation that I downloaded, which is the lung lower lobe left. Again, change it to segmentation, and yes, you see, this is the lower lung lobe. So you can manually download these segmentations and keep uploading and see, if it's the right structure or not. So yeah, you may want to play around with it a bit. Any problem running total segmentator and downloading it and then visualizing in 3D slicer? You can do the same thing. Um, you can get it to the middle. Oh, first I'll have to do show 3D. Get it to the middle. Uh, Run this, and then remove all the axis and everything. Make the background white. A And then start spinning the aorta. So this is the aorta looks like. It comes out from the heart and then goes all the way, like, towards the dorsal region, and then the abdominal aorta bifurcates into the iliac arteries. So while working with total segmentator, I learned a lot of anatomical terms. Okay. Any problem? No, right? Okay, so you can try uh, with MRs. We can try to visualize it later. Uh, Let us move on to the next topic, which is uh, training your own NN unit. So till now we were all using already existing segmentation models, right? But now, let's say you go back to your institutions and then you are working with a new data. So let's say you are working with, let's say, some segmentation which is not there in total segmentator, or you got your own data which has the lung CT and the tumor segmentation or whatever, depending on your task. You have the image and you have the segmentation. You want to train your own segmentation model because none of the existing segmentation model does that. So here is a very brief tutorial. Like, believe me, you don't have to do anything in this if you use this. Uh, just you have to pre-process the data and you can train your own segmentation model. You can train your own NN unit. NN unit was published in Nature Methods in 2020, if I'm not wrong, and it became one of the state of the art method for segmentation. You can actually, like, it's a very good paper. You can read this paper maybe uh, later, 2021, and it was published in Nature Methods. So it's basically NN means not a new unit. So it's unit, but with a lot of hyperparameter optimization and automation. But like NNUnet outperformed all the existing segmentation baselines, and it's pretty accurate for any task, I would say. So if you have an image and if you have a data set where you have the images and the segmentation files, you can use NNUnet. And how to use it? I have compiled this code. It's very easy code. Just change the path and everything, and you can train your own segmentation. So first, you'll have to install NNUnet, and then, as of now, I have downloaded. I use this data, but I'll tell you how to structure the data. So you can also take a look at the NNUnet GitHub. It's very informative. The NNUnet GitHub page. Here they have very good documentation about how the dataset structure should look like, etc., etc., how NNUnet works. If you have a 2D dataset, for example, if you want to segment the lungs from chest X-ray, you can use NNUnet. If you want to use this to train CT, like segmentation for CT, you can do that. So it's like very like generalized in that form, that manner. Yeah. So it supports PNG, TIFF, NII, NRRD, all the different imaging formats that you can have. So how you should keep your data? For example, like you should have a NN unit raw folder. Mind you, the U and N should be capital. You can't mess up with this capital and small. So you should have a folder like NN unit raw, and in that you should have different data sets that you want to train in this format. Dataset underscore something 3-digit number from 001 to 999 underscore any name that you want to put. So this is not standardized. This is your name that you want to put, but this one should be same. So for example, you see like the data set same for everything, it's just the number changes, and this number should be unique. Like for example, today you are trying an experimentation data set 001 underscore, let's say, tumor segmentation. Tomorrow you are trying to redo another experimentation, don't put data set 001, just put it something else, 711 or 911, something like that. So inside this data set 3-digit number underscore something, this is how you should keep your data set. Images tr, that means images for training. Labels tr, that is labels for training. So in the images tr you will have your uh, the image files, and inside labels tr you will have your segmentation files, and a dataset.json, and images.ts is optional. That's all. And mind, like this T should be capital in TR, and this T should be again capital in labels. And inside this, you see, you should have all the imaging files, and inside labels, you should have all the label files. Okay, so let's see. Restart session. I will run this. I will download this dataset. So after you have formatted this dataset in this format, what you should do is you should just run this command. And this 106 is basically your dataset number, which is this one. This one. That's it. So if you can keep the dataset in this file folder structure without messing up with the capital and small letters and the underscore and the number and everything, you just have to run this line. And this 106, I'm sorry, this 106 changes to whatever the dataset number you keep, 711, 911, like it's totally depends on you what you want to put, and then run it. So what this will do is this will automatically pre-process the data. So you remember yesterday we were talking about normalization, bringing the pixel value up to some range, this like multiple things. You don't have to do that anymore. NN unit automates everything. So NN unit is, I would say, particularly built for clinicians who want to use segmentations but don't want to write code. I mean, they don't have the time or bandwidth or the interest of writing this dense normalization and pre-processing codes. So it's very useful for them. Just keep the images and the labels in that particular file format, run this command, have a good GPU, start your segmentation training, and it's very accurate. And after that is done, this is your training command, just one line. NN unit underscore train 2D NN unit trainer v2 task 106 multilang 0. This 0 means 0 fold. So if you want to train it for 5 folds, just so that is basically this dataset that I downloaded. I can show you. So here the dataset that I downloaded already named it as 106. You can name it anything for your dataset. So for example, I was initially trying to do this with Brad's dataset, and so this is what I was trying to download, but it was so large, and the internet was was a bit slower. So this is how I kept my Brad's 2D dataset. Can you see this folder? Dataset 143 Brad's 2D, and inside this I have a dataset just .json and the images tr file and the labels tr file. Inside the images tr I have all this brain MRI scans. Inside this label I have multiple labels. So it's black because it's 0 to 1. You can't see it with naked eye, but the labels are there. And then this is the dataset.json, where you just say that I have this different MR sequences, flare, T1, T1 post contrast, and T2. Two things I want to segment: background, which is anything that is black, and the tumor, which is anything that is white. And this is the number of training samples, 776, and the file ends with .png. So this is how your dataset .json should be, and then you are good to train. So I have a question. Why labels tr has 776 items and image trs has almost 3000 items? Sorry, no, it's not augmentation. So uh, you see, like for tumor segmentation, I was discussing yesterday that you need 4 different sequences of MR: the flare, T1, T1 post contrast, and T2. So how NN unit basically trains itself is, it takes all the 4 sequence, feed it to the unit structure that it has, and provides one mask output. So for example, if we have this label, let me view as list. So if you have this patient Bratz 2021 underscore 00016 PNG, for that we will have 4 Bratz 2021 underscore 00016 files. So what are these 4? The 000 is, if I go to the dataset.json, is my flare. The 001 is my T1. The 002 is my T1 post contrast, and 003 is my T2. If you have multiple sequences for the same patients, you name it like this: underscore 0000, 0001, 0002, and 0003. So for each label there are 4 MR sequences. So coming back after this pre-processing is done, yeah, I hope this pre-processing is done. I'll just hide these 5 different folds and I will start running training. Yep, it says I'm running an unit 2D. There's only CT, batch size of 104, and creating a new 5-fold cross validation, training fold 0. There are 15 training, 13 in validation. LR learning rate is 0.01. Just give me a warning that you are trying to train on CPU. Doesn't matter. I'm not going to train it till end, but yeah, you should use GPU. I'm just trying to wait for one epoch. But this is how you can just train your own NN unit if you have the correct file folder structure. So segmentation is very easy. Any questions? So what I would suggest is I will share, like after the session, of course it takes time. After the session, I will share a BRATS, that 2D folder that I'm trying to download with you. You can, I'm not anything. You can try to run your own NN unit code for the BRATS tumor segmentation. Like after the session is done. Yes. Microphone.
Sir, learning rate is 0.01 you given. If it is I'm increased to LE4 like 0.001. So what are the consequences we can face that one? Are you observe anything regarding to this one?
Okay. So I can answer this question in 2 phases. First, I don't decide the NN unit 0.01. So this phase, you see, like this NN unit plan and pre-process does not only pre-process the dataset, it also plans the training. So based on the dataset, it starts with 0.01, but after every iteration, it can change the learning rate. So now it's, let's say, 0.01. Next, it can, like in the next iteration, it can go to 0.001. But so NN unit is very much automated. That's what I was saying. You don't have to do anything. You don't have to train multiple times. But again, second phase of my answer is, I have seen that if you have a very small data set and if you want to train that data set, like with of course you want to have good accuracy, but the data set size is small, and to get better accuracy you want to train a bigger model. So model is big and the dataset is small, so you would like to train the model very slowly. By slowly I mean the learning rate should be decreased. So so rather than keeping 0.001 or 0.00001, you can keep it like up to 5 decimal points, 0.00001 or 0.00001, something like that. But you should not train. So for example, if you are planning to train your model for 500 epochs, you should not keep the same learning rate. What you should do is you should put a decay. That means, let's say if you started with 0.01, let's say even for smaller data sets you would like to start with like higher learning rate, so that the first few iterations the model learns faster and then you slow down. So for example, for the first 50 epochs you train with 0.01, next 50 epochs you train with 0.0001, and then the next 200 epochs you train with a very lower learning rate. And so that is called weight decay. You should also have something called the scheduler. So that will check. So scheduler is basically, I mean, it goes into again, model optimization stuff. So let's say if you have a convex function and if the goal is to reach the global minima, and if you have the step size which is very big, that means the model will never reach the local minima. So you should like slowly gradually reduce the size. So if you're on the top of the hill, you would likely to come down fast initially because you are not even near to the global minima. So have large steps, and then the more you reach the the global minima, reduce the learning steps, because if you don't reduce the learning steps, even if you are here, you will still go here and there because the step size is very large. So learning rate, scheduler, and then also what you can have is early stopping. If your model is not training anymore, just stop after 20 epochs if the loss is not converging. But again, coming back to that, there is no, like, how to say, correct answer how to train a deep learning model. You should have all these recipes, but again, like, that does not guarantee that if your training is very difficult, you will get the best accuracy. It's all about, like, training. They have a good model, like big model, big data set, everything. Okay, let's see if we are there for one epoch. No, I mean, of course, we are training on a CPU, so it will take time. But yeah, if like, I know, and running an NN unit in Colab is difficult, but you can take this code if you have a good GPU in your lab, or if somebody is nice enough to give you GPUs for training. You can contact Samyajit. So yeah, uh, yeah. So that is basically the summary of this lecture. We'll do till 5:30, so I have 12 more minutes. Please bear with me. I know it's Sunday, but I have just one topic left. Very small topic. You don't have to do anything, you have to just listen. Yes. Yeah, any question? Sorry. The NN unit, I will send everything at the last, uh, because I have to compile, sit down and compile. And to be very honest, I'm not getting any time between. Sorry? Oh, just type NN unit. Just type NN unit. Don't mess up with the spelling, and the first thing you will get is NN. Okay. So why I want to emphasize, because this is the last and I would say one of the most important thing if you're trying to do some project. Yesterday I discussed radiomics. And if you listen to the lectures of Dr. Prasanna, he was emphasizing on this radiomic features and then doing all the different downstream tasks using radiomic features. Now, if I tell you, and also I discussed TCIA, you have those huge data sets, and I think you had a question that how can I download this data set? Well, pathology data set I can't help, but at least if you're trying to work with radiology, I have a good solution for you. So these datasets are huge. Like, if you try to download these datasets, it will be in GBs, and then processing it will need time, and it will, like, you will get multiple errors, and at some point you will just dump it. But recently there is a link which is called Radiomix Hub. What they did is very nice of them. They took all the datasets that you need, LIDC, IDRI, non-small cell radiogenomics, non-small cell radiomics, UCSF, UPenn, etc., and, like, downloaded those datasets, pre-processed them, and provided you the pyradiomic features. So you don't have to do anything. Just select what research you want to do. You want to do work on lung cancer, take these two datasets. You want to work on prostate cancer, take this dataset. You want to work on head and neck squamous cell carcinoma, take this dataset. You want to work on kidney, take this dataset. You want to work on liver cancer, HCC test, take this dataset. So you don't have to download this dataset anymore. Click here, you get all the radiomic features extracted. And not only the radiomic features, also their labels. So for example, this patient, HCC001, is it dead or alive? Dead. What's the overall survival? This one. Did they have hepatitis? HCV only. What was the age? 71. What was the gender? Sex? 1. Smoking? 1 means yes. Alcohol? 1 means yes. So you get all the patient history and their radiomic features. So pyridiomics tutorial that I gave you yesterday, you don't need that anymore. But still, if you get your own data, you still need to run the pyridiomics, and I hope it was very easy, just few lines. But if you want to work on some public data sets, just save your time, take the radiomic features from here and run your classification model. Now, how to run your classification model? Yes. Sorry, any question? No? Okay. So I downloaded the non-small cell lung cancer radiomics, that is basically this dataset. And here, of course, there are all the radiomics features like SAPE, texture, GLCM, and all those things that I discussed yesterday. So I downloaded that, and I have all the pathology information, like what's the age, what's the clinical stage of the tumor, uh, and the histology. That is what I'm talking about. So if you see the histology, there is large cell, there is squamous cell carcinoma, and there is adenocarcinoma. Now let's say from this set of patients we want to see if these radiomic features are predictive of classifying squamous cell carcinoma and adenocarcinoma, that is LUAD versus LUSC. So this is a very simple code, but let me first discuss the pipeline. Let me ask you a question. So there are, sorry, so there are almost, like, I don't know, like maybe more than 100 features here. Do you think all of these are important? No. I think Dr. Prasanna also discussed that not all the features are important. So how would you know which features are important? So you have to do something called feature selection based on ranking. So the feature selection can be done like using various techniques, but like ranking them is one of the, using ANOVA, which is a statistical method, is one of the best approach to do. So here I just download them, load it here. Let me load it again.
Excuse me.
Yes.
When you say ANOVA,
Yeah.
what form of ANOVA? Any form, 2.3 point, anything does the same work?
I don't think anything does the same work. I think what I take is the most recent one, which is ANOVA V2
Okay.
or ANOVA V3. I have not used V3.
Okay.
Mostly we use. It's not about me using. I use this fclassif, uh, this sklearn library, and then here I have this fclassif function which uses ANOVA. I think if I'm not wrong, they uses
Inbuilt class. Okay.
inbuilt.
Okay,
Yes.
I get.
Yes. Any more questions? No. Okay. So let me try to upload. I have downloaded that in my local system. So let me. Okay. So my features and labels both are uploaded. So let me run it one by one. So here what I do is, so if you see this radiomic files, there are some, like, columns which are not radiomic features, but it has some information. For example, the ROI, image path, the segmentation path, and the extraction ID. I don't need these things. So what I'm doing is I'm just like dropping those columns, dropping those, and from the label you can do multiple things. You can do survival classification, blah blah blah, but here we are doing subtype classification, squamous cell carcinoma versus adenocarcinoma. So I'm only interested in this histology column. So what I do is I first calculate how many patients are there for which. So for squamous cell carcinoma we have 152 patients, for large cell we have 114, for adenocarcinoma is 51. So I just remove the large cell and NOS, and I'm just taking this 152 and 51 patients. So yes, I do it here, and then my target class is adenocarcinoma and squamous cell carcinoma only. So that becomes this 202 patients, and then I map the class. So adenocarcinoma is labeled 0 for me, and squamous cell carcinoma is labeled 1 for me. Then I drop the unwanted columns. Now here I built a pipeline. So as I said, first thing is you have to standardize the data or normalize the data. For that I use an inbuilt function called standard scalar. Then what I think Salik was asking, select the best k using ANOVA, which version of ANOVA. So this is what the select best feature is happening here. And then finally is the classifier. So normalizing, selecting best feature, and then classification. This is the pipeline. So my random forest is one of the, the best classifiers that you can use. You can also use support vector machines or decision trees. You don't have to write code for anything. They are already there in sklearn. So you can just pick and play with these classifiers. But what I would suggest is, don't spend too much time, like, experimenting which classifier is working the best. For that we have another library called Optuna, which does this hyperparameter tuning or hyperparameter optimization. So what is hyperparameter optimization? So you want the best model. and you don't know what your best model can be. But you have a fair idea that it will be somewhere between, or it can be somewhere any model among support vector machine, logistic regression, decision tree, or random forest. So you want to feed this uh, to the hyperparameter optimization framework, and let it pick which one will work best for you. And if the model selects SVM, it does not know what the sigma function, the gamma parameter and the C parameter will be. So all these things are done by hyperparameter optimization function, that is Optuna. So you don't have to worry about, so it will finally, the final output will be the best model you can have. So I'm downloading Optuna, and then I'm running this hyperparameter optimization for only 20 trials. So here, rather than keeping 20, you can do 500. So like the more the number, the better search it will do, the best parameters. So as of now, just for time constraints, I'm doing 20. I'm already at 13. So you see what's happening here. In trial 0, the value, my AUC is 52%, which is not good. I don't like it. And the best features were 24 features, 24 top features were selected. We used a random forest model with so and so parameters, but I don't like 52 AUC. I want more. So the next iteration, trial 1, what happened is it changed from random forest to SVM. The Optuna is doing it in itself. I'm not doing anything. And I increased the number of parameters from, number of best features from 24 to 48, and it came 54. I don't like that anyway. So you keep increasing, and somewhere you have so this is the best configuration with best parameter, like the top parameters 90. Logistic regression was chosen the best model, and this is the log reg C value. And yep, you see my test accuracy is 60%. So I started with 50, 52, and now it's 60, and AUC is 52. And then you can see like adenocarcinoma, the precision is 33, squamous cell carcinoma, the precision is 83. Why the model is predicting squamous cell carcinoma better? Any idea? It had more number of patients. So it's a very imbalanced data, and that is why the model is overfitting on squamous cell carcinoma, which is not a good thing. But there are other ways to deal with class imbalance that we'll discuss later. But yeah, summarizing, uh, you have Radiomix Hub from where you can get all these features. You don't have to download the image, you don't even have to see the image anymore. You have all these Radiomix features, and you can use this entire pipeline end to end to do a classification. And also you don't have to burden yourself with selecting the best model, trying to improve the accuracy. This hyperparameter optimization library is going to do that for you. So at the end, you will have the best set of results that you can have.
So a lot of effort has gone behind preparing these codes. Okay, so please take this opportunity to learn, and I hope uh, upgrade your skills as well. You have come also from far places, so it's time to actually, in addition to enjoying the nice winter weather, please do take benefit of these things. Anyway, so I think with this we finish our experimental session. Thanks to Moinak for all the explanation and support, and I hope you all had a good session today.